start by discussing the primary structure of proteins and then work our way up, okay? So the primary, bottom structure of a protein is just the amino acid sequence. So the bonds we're looking at this level are just peptide bonds. Remember, peptide bonds are a type of covalent bond, and they are bonds that hold one amino acid to the next. Now this might not seem super special, but remember there are actually about 20 different amino acids that can appear in any order, right? And just a single difference in this sequence will produce an entirely different protein. So for some perspective, let's say we have a protein that's 100 amino acids long. This would mean that there are 10 duoquadragentillion ways that amino acid could be arranged, meaning 10 duoquadragentillion different proteins can be made from a sequence of just 100 amino acids. For some perspective, that's multiple billions of times more than the number of grains of sand found on the entire Earth. It's this property actually of proteins that allows there to be so many different proteins, each with their own unique shapes and functions in our bodies. So again, the primary structure of proteins is just the amino acid sequence, meaning at this level, we're just talking about amino acids, what order they're in, and their peptide bonds. Now let's look at secondary structure. Secondary structure has to do with how different amino acids along the polypeptide chain interact with one another. So at this level, we're looking at hydrogen bonds. When we are looking at the primary structure earlier, we were just looking at how one amino acid is linked to another. But here, with secondary structure, we are looking one level beyond that. We are looking at how amino acids along a chain are basically intermingling with one another. And this contributes to the shape of the overall protein. The primary structure goes over what amino acids are present and in what order, but the secondary structure is the first level to help determine the shape, and therefore also the function of the proteins. So let's explore what this means. So drawing out our primary structure, like so, where each different colored ball represents a different amino acid. The secondary structure is when we have amino acids along the polypeptide chain interacting with one another. So if you can imagine, these interactions and hydrogen bonds forming between the amino acids are located in different parts of the chain, and this causes the polypeptide to form different certain shapes, kind of like so. See how the bond is holding these two amino acids closer together within the chain now? Yeah, because of this hydrogen bond between the two amino acids, we have a bend form in our chain. Our polypeptide is taking on a specific shape. So there are two common shapes we find in secondary structures of proteins, thanks to these hydrogen bonds. Now I do want to clarify, the amino acids in this chain contribute to which amino acid is interacting with another amino acid. So does the primary chain also influence shape to a degree? Absolutely. It's only in the secondary structures that we see that shape starting to take form, okay? Now let's explore the two common shapes we find in secondary protein structures. Our two structures are beta pleated sheets and alpha helices. Now, beta pleated sheets are a planar structure, meaning it looks kind of like a flat, ziggy-zaggy structure. And these sheets form because of where the hydrogen bonds form between amino acids along the polypeptide chain. Now, alpha helices, on the other hand, as their name would imply, are in the shape of a helix, or a coil, right? So imagine you have a line of amino acids, like so, trailing off the screen, and amino acids form hydrogen bonds with one another along the chain, like so. Well, these bonds cause the chain to take on this kind of helical shape. To help you see how the chain goes from a line to a helix, I'm going to go ahead and number off and mark up some of these amino acids here to make it easier to see, okay? So here we go. Here's how it's going to change. There. Can you kind of see how the hydrogen bonds pulled them together? Now, I sometimes find interacting with 3D models to help with visualizing these secondary structures. So if you'd like that option, you can actually do so in the links, through the links that I embedded in your course content for this section. All right? Pretty fun stuff. Highly recommend if you're interested. All right? Cool. And now with that, let's have a brief check-in. Check-in 24. Make sure you're looking at number 24 now on Canvas to answer this question. Fill in the blanks. The primary structure of a protein is looking at, specifically at the bonds between amino acids. The primary structure does or does not influence the shape of proteins. You can or cannot see the protein taking shape in the primary structure. The secondary structure of a protein is looking at bonds that form between amino acids along the polypeptide chain. The secondary structure does or does not look at the shape of the protein. All right, now let's look into our tertiary structures. Tertiary structures largely refers to the interactions between or among many secondary structures. Basically, we are now talking about how many secondary structures are arranged and how those structures associate with one another and with their environment to make one large globular shape. Let's start by looking into interactions with the environment first. The environment that components of life exist in is what we call an aqueous or water-containing solution. So let's imagine this is our environment surrounding our protein, just full of water. And in here, we have our secondary structures of our protein. Now remember, each amino acid within a protein has their own special R group, or side chain, which cause each amino acid to have unique chemical properties. Now, as you can see, I'm only writing a few R groups in here, but remember, in real life, all amino acids have an R group. One of the properties of these R groups, or one of the properties these R groups can carry, is they can be polar or nonpolar. Now, remembering back to our discussion on water, is water polar or nonpolar? It's polar, right? Right. So other polar molecules are attracted to water. We call these molecules that are attracted to water hydrophilic molecules. Hydro meaning water, and philic meaning attracted to. I'm letting these blue-colored R groups represent our polar hydrophilic R groups, okay? In contrast, our non-polar R groups that I color-coded in red here are groups that would not be attracted to water. We call these hydrophobic. Hydro, again, meaning water, and phobic, meaning to fear or dislike. So if it helps you remember, you can think about phobias, right? Such as claustrophobia, which is the fear of tight spaces. So things tend to avoid their phobias. So, as you can imagine, if you have a bunch of R groups that are nonpolar hydrophobic, they will associate and attract to one another and basically try to avoid water, while hydrophilic R groups will interact with the surrounding water. These interactions basically cause a secondary structure to come together to form a glob-like structure, where the hydrophobic portions of the structure are on the inside of this glob, associating with each other and away from water, while the hydrophilic portions of the structure are on the outside, interacting with the water. Here's actually a GIF I like to show that sort of shows the hydrophobic amino acids getting internalized within the forming protein, while the hydrophilic amino acids are on the exterior, interacting with the surrounding water. Now let's explore how different secondary structures interact with one another in their tertiary structure. Once the secondary structures are basically smushed together, there are a lot of different ways that the strands can interact with one another. For instance, we have our standard hydrogen bonds. But we also have something called disulfide bonds, or disulfide bridges, which forms if two amino acids have cysteine R groups. Cysteine R groups are basically R groups that have a carbon and two hydrogens along with a sulfur. The key is sulfur. And a bond can form between those two sulfurs, linking those R groups and then those two amino acids together. And we call that a disulfide bond or disulfide bridge because two sulfides. Another common bond formed in the tertiary structures are ionic bonds, which form between groups that are oppositely charged. So if you have an R group with an overall positive charge and another R group with an overall negative charge, kind of think of it like magnets attracting each other. Those are our ionic bonds. And the last common molecular attraction we find here are van der Waals forces. By themselves, van der Waal forces are actually pretty weak. But if there are many, many, many of them in close proximity, these forces do add up and can actually become very strong attractions if there's enough of them. So, with that out of the way, let's clean up this drawing a bit and zoom out. Here is a more zoomed out view of what a tertiary structure of a protein could look like, where we have several secondary structures folded and bonded together interacting with one another throughout the structure. Now, at this level, our protein is not functional yet. Our protein is only functional once it's reached its quaternary structure. So we're almost there, but not quite. So let's talk about our quaternary structure. So just as a brief review first, for our tertiary structures, we are looking at interactions between and among secondary structures and their environment, which specifically includes our hydrophobic exclusion, where we have the hydrophilic portions of the polypeptide interacting with water, and the hydrophobic parts all coming together on the inside of that structure, avoiding water together and interacting with one another. We also have hydrogen bonds, our disulfide bonds, ionic bonds, and our van der Waals forces. All right, now let's move on to our quaternary structure. So, in short, if this is a tertiary structure, then this, all of these parts together, assuming they're all intermingling, they're all connected and bonded together somehow in different parts and different areas, they're all interconnected, these parts all together are our quaternary structure. The quaternary structure is basically the arrangement of several tertiary structures, each a subunit to the overall protein, all together. The bonds and forces that come into play when we're looking at tertiary structures also come into play here. But in addition to there being interactions between secondary structures and their environment, now there are also interactions among tertiary structures. So several of those as our subunits coming together, making this quaternary structure. This here is our fully functional protein. And while its shape might look a bit random, it's actually not random at all. The shape of this protein is very, very, very highly specific. So specific that if even one amino acid somewhere in this entire thing is altered, then the entire shape of this protein would change, rendering this protein to be non-functional, meaning it cannot carry out its role in the cell. It wouldn't work. It'd be a broken protein. All right, so with that, let's circle back. Here is our review of what we covered in terms of the levels of protein structure. These are the big takeaways. But the other big takeaway from here is that at each and every level, the order the amino acids are in throughout the polypeptide, as well as the shape of the polypeptide at each level, are highly, highly specific. And if there is any alteration, no matter how small, the protein will not be able to function. Shape is directly related to function, and the order of the amino acids determines the shape. So if we're looking at our peptide bonds in our primary structure and the amino acids in their order, the order in which they are in determines where hydrogen bonds will form along that chain, right? So the order is helping determine this level of shape. And then this level of shape, the secondary shape of our polypeptide, determines the tertiary shape, and the tertiary shape helps determine the quaternary shape. So if there's a breakdown in any of these structures, then the entire protein will not function. Make sense? So circling back to here, feels like a bit, right? We're back. We've now gone over amino acids. We've gone over levels of structure. Now we're going to go back into how shape is so integral to function. To do this, let's look back at what the main functions of proteins are, and then look at just a few examples for why structure is so important to function. Starting with reaction facilitators. These type of proteins are often called enzymes. So if you've heard enzymes before, yep, we're talking about proteins. And their shape is incredibly important because they must be able to bond to the molecule or molecules, can be multiple, for which they are facilitating a reaction. Let's take digestion, for example. Yeah, let's say you're eating a piece of bread, and that piece of bread obviously has a lot of complex carbohydrates, right? So complex carbs, a bunch of sugars linked together, a polysaccharide. But the human body cannot actually use or absorb polysaccharides. They must be broken down into their monomers or monosaccharide units first for us to be able to use it. This breakdown is facilitated by a protein, an enzyme, that binds to the polysaccharide and breaks down the bonds along the chain to produce those monosaccharides. Now, if the protein were a different shape, it would not be able to properly bind and therefore break down those bonds within that polysaccharide. And so we wouldn't have those monosaccharide units formed from this. It would remain a polysaccharide. This same relationship that shape facilitates the function of the protein is also found in the rest of the examples, but running through just two more really quickly. For defense, right? We have antibodies. You've probably heard a lot about those recently. Antibodies are a type of protein which bind to foreign molecules, such as a virus particle, right? If the antibody is the wrong shape, it will not bind to the viral particle. It can't. It doesn't fit. And if that happens, then the antibody can't aid our immune system. if a protein is responsible for transport or storing molecules, it must first be able to perfectly bind with that molecule. If the shape is wrong, then this can't happen, right? And therefore it can't function. Now you might be noticing a pattern here. In each of these examples, the protein is binding to something, right? So we have a protein that's binding to something. And this is usually the relationship. There are a couple of exceptions, such as when we're looking at collagen, for example, but largely this is the relationship we see, where something is binding to something else. So this something that's binding to proteins is often referred to as a substrate. So if you hear the word substrate in the future, that's all we're talking about. We're talking about what's binding to our protein. A lot of people like to refer to the relationship between proteins and their substrates as sort of a lock and key mechanism. In truth, it is actually a lot more complicated than just that, but if this metaphor helps you, go for it. It's not really a bad way to think about it anyways. Now, like we've said so many times now, if a protein is the wrong shape, right, it cannot bind to its substrate, and it therefore can't carry out its function, right? It can't do it. If a protein is the wrong shape, though, due to environmental factors, we call that protein denatured. So if you hear the word denatured, we're talking about a protein that no longer functions, right? It's the wrong shape. Some environmental factors that can cause denaturation are when a protein is exposed to, let's say, improper pH environments, also improper temperatures, or poor ionic concentrations surrounding the protein. All of these can lead to denaturation, right? And if a protein is denatured, it does not function. All right, check-in 25. This is a search and apply question. These are kind of fun. So this is where you are going to read a prompt, you're going to be asked something, and then you're going to go search for the answers using what you learned from this lecture, as well as outside resources to help answer the question. Sound good? Okay, and this is purely for educational purposes. This should be a fun educational endeavor. This is not stressful. If you need help though, always feel free to reach out, okay? I think this is really cool. So let's dive in. A person possesses a mutation that impacts the amino acid sequence in hemoglobin. This mutation causes a single amino acid, glutamic acid, to be replaced by an amino acid called valine. First, would the single amino acid change have an impact on the human? What do you think? If you believe this would have an impact, go ahead and look up what that impact would be. I think you'll be surprised. All right, and if you learn anything interesting while on this endeavor, feel free to share that too. Sound good? Okay, best of luck. And that concludes our introduction to proteins. Awesome work!